Hi, I'm Jacob Adillo, and we're going to be talking about DNA replication. Okay, so the parent strands are unwounded by the DNA helicase, which splits into two individual strands, which takes place in the replication fork. You may ask yourself, what is the replication fork? The replication fork is the point where the DNA will be split and the new created DNA will be synthesized. Well, after that, the single-strand binding proteins stabilize the unwound template so it would not snap back together, and these are binded loosely. Okay, the enzyme primase uses the original DNA sequence as a template to create shorter RNA primers. This is important because DNA polymerase cannot create its own nucleotide chain, but can only extend it. This makes primase very important in DNA replication. The DNA polymerase synthesizes a new DNA sequence which is extended by the RNA primer. This starts at 5' and goes towards the 3'. This will prompt up DNA, which will create a complementary strand. Since the DNA strands are anti-parallel, this means the polymerase are going to move in opposite directions, which are held by sliding clamps, which allows the DNA to be recessive. Due to the nature of DNA, only one of the polymerase is going to be able to copy the DNA continuously in one direction, and this is referred to as the leading strand.
We just got a letter, we just got a letter, we just got a letter. Wonder who it's from.
It says here, the other polymerase is classified as the lagging strand, which could only copy a short strand of DNA before it goes through the previous primer, which causes the lagging strand to repeatedly release the DNA strand to slide up further up to the next DNA primer. In the lagging strand, the DNA parts that were separated by the DNA primers are known to be called the Okazaki fragments. Okay, so we are going towards the end of DNA replication. So in the lagging strand, what needs to happen is that we need the lagging strand to be able to take out the RNA primers as well as bond the Okazaki fragments together. For this to happen, something called the RNase H needs to occur. So what happens is it degrades RNA primers, which creates a gap for the DNA polymerase to fill the 3' end of the neighboring Okazaki fragments, and are finished off when the DNA ligase binds together one of the 3' end of an Okazaki fragment with the other 5' end, and this takes place within an ATP or NAD dependent reaction.